So you have the best press releases recently. Solid tumor, cell therapy, advanced melanoma, advanced cervical cancer. If anyone hasn't heard about these developments, then they should probably leave the room right now, but just to save them that embarrassment, give us the update on LM145 and lifileucel and where exactly Iovance is in their trajectory.
Okay, so Iovance Therapeutics is a company that manufactures tumor-infiltrating lymphocytes for solid indications. So basically, in short, we take a tumor biopsy, we have a process or processes whereby we create an environment for the tumor-infiltrating lymphocytes to emigrate out of the tumor, and we get them to expand, and then we adoptively transfer them back into the patient. So it is a polyclonal heterogeneous population of cells that is specific to the patient. We have several ongoing clinical trials, phase 2 clinical trials currently in progress. We have LN144 for metastatic melanoma, which is also referred to as lifileucel, as well as LN145 that we are using to treat metastatic persistent cervical cancer. In terms of our pipeline, which we'll probably get into, we have other things that we're working on. The press releases right now is that we recently talked about our most recent clinical results in metastatic melanoma, where we saw an overall response rate of over 36% in this highly refractory population, stage 3, stage 4, that have exhausted all other treatments, including checkpoint inhibition. Even more interestingly, we've seen in cervical cancer, highly refractory patients, again, chemo-resistant, and we have seen an overall response rate of 44%. What's interesting is not only have we seen responsiveness in both of these populations, we have not reached something called the duration of response. So in our metastatic melanoma cohort, we have reached out to approximately 15.5 months, and we still see that these responses are ongoing, showing that they are indeed persistent and durable. We have other things in the pipeline. We're treating patients with head and neck cancer. That's a fairly new indication that we're looking into. We're also getting a little bit into liquid tumors with CLL in a program that we have with Ohio State. Some of our other press releases recently has been some collaborative relationships with Novartis, where we have licensing now to produce an IL-2 analog. Part of our process involves systemic administration of IL-2, which can bring about some significant adverse events. And so we want to try to decrease those adverse events in our patient cohort. We also have initiated a collaboration with a company called Selectus for their TALEN technology with the purpose of genetically editing our TIL, because currently our TIL product is not genetically engineered. It is expanded from the actual tumor biopsy itself.
So it's not hard enough to just manufacture TILs, you have to gene edit them as well to introduce further manufacturing challenges?
Yes. I think you can look at it in the perspective that in the pipeline we're looking at multiple types of strategies to try to improve our current product. So we've seen dramatic and remarkable clinical efficacy in the indications that we've looked at. We want to expand that repertoire in terms of the indications that we're treating, some that may be even more difficult to generate TIL. And we want to know that we can actually produce a population of TIL that is persistent and durable.
No, your clinical numbers are reminiscent of nothing more than the trials like Eliana in the earliest days of CAR-Ts, where we saw crazy numbers in the 80% range. There is no mistake that these numbers look amazing. Let's talk a little bit more about the manufacturing. You start out with a tumor biopsy.
Correct.
And you have manufacturing operations lasting over 20 days.
Correct.
How do you feel about the robustness of that process?
So the initial process, which was devised by Steve Rosenberg Labs at NCI, 30 years of development with this process, initially was a 6-week manufacturing process. In terms of the ability to commercialize the product, it was going to be very difficult to do so, to manufacture something that was going to take that long in manufacturing. So one of our first goals at Iovance was to be able to come up with a process where we could expand the TIL and in sufficient number to see clinical efficacy, but do so in a much shorter manufacturing process while cryopreserving the product. So one of our first initiatives that we generated is we call it the Generation 2 process. So it is a 22-day process. And on average, we're able to expand TIL from a tumor biopsy to approximately 28 billion, that's on average, 28 billion cells, and still see fairly significant clinical efficacy.
22 days, still a pretty long manufacturing process for autologous medicine.
It is when you have a patient population that we're currently treating. We're looking at patients that essentially are towards the end of their life. And so 22 days is still a long process. So we are currently working on further optimization of our process, and we're trying to reduce the number of days while not affecting the product's efficacy. That's important in terms of cell number and the cell population that we are deriving.
How manual is the process?
I'm sorry?
How manual is the process?
It's actually a fairly streamlined process, so much so that when we were first working on it, we had to have everybody in the laboratory involved to make sure that every single person was able to do so. Right now we are actually building our own manufacturing facility that should be ready in about 2021, 2022. Right now we have manufacturing facilities generating our TIL product for us, so we have a technology transfer team. And so the biggest undertaking is at day 0, but it's just basically, we do it in a very minimalized culture system whereby basically you fragment the tumor, you throw it into a flask, you leave it for 11 days, and at the end of that 11 days, we add in something that causes rapid expansion, and then at day 16, the cells are split into multiple flasks, and day 22, you harvest. So it's a fairly simplified process compared to the initial process where it was involving 24 wall plates and multiple plates and constantly splitting the cells.
Still sounds really expensive.
Yes.
How much?
That I can't necessarily speak to. I can tell you in terms of the fact that we have a
are we talking Kymriah expensive or Zolgensma expensive? Or maybe more?
You know, we're still working at the logistics of how much it's going to cost. The thought process in having our own manufacturing facility and being able to tweak the process is to try to make it as accessible to as many patients as we can. Being that the fact that right now our current product is not genetically engineered, and it is indeed just expanding cells from a pre-existing tumor biopsy, and doing so with really only 3 days of cell culture, we're hoping that it's going to be a reasonably derived product in terms of time and in money.
So if you fold in gene editing, it's going to be even more expensive?
Likely. But the thought of gene editing is further down into the pipeline. Again, with the gene editing approach, we're trying to utilize an approach with Selectus and with the talent whereby we're going to do as little manipulation as we possibly can, but very much we're still in the preclinical steps of that particular process.
Yeah, I mean, you understand the sensitivity here. We saw the a very manual Kymriah process, which is, to be polite about it, has faltered on numerous occasions, and then we see this inflation, this apparent ongoing inflation as we move up through Luxturna, just under 1 million, Zynteglo we'll see, and Zolgensma over 2 million. You could almost buy a house in the Bay Area for that money, but probably not quite.
Our hope is to be that it's going to be more affordable than what we're seeing currently with some of the cellular immunotherapy.
More affordable than CAR T type cell therapies?
Yes, that's the hope. Now our current product, as I said, it's an unmanipulated product. And again, we talk about further generation to try to make it more potent, more anti-tumor. But one of the strengths of our current product is that it's unmanipulated. Mm-hmm. And we think that that's the strength when you're thinking of solid malignancies versus liquid tumors, and you think of the evolution of CARs versus TIL, right? If you look from the perspective of a solid tumor microenvironment, you have many, many tumor antigens. It's a very heterogeneous population. if you look on average, there's potentially thousands of TCRs, 20,000 we've been able to see of TCRs that are present within a tumor lesion. You could think of the number of tumor antigens. We really don't know what tumor antigens or what T cells are tumor reactive. Even with the evolution of whole exome sequencing, RNA sequencing, the identification of neoantigens, the strength of TIL is its polyclonality. In terms of CAR, it's difficult because CARs are typically to one antigen. They work very well in heme malignancies.
One cell, sometimes,
Yes.
if you believe Bruce Levine, all you need is one cell. If only we only need to manufacture one cell. I think that is the great strength, and there's two sides to every coin. What worries me about manufacturability is when I hear about the polyclonality of a product. What excites me about the patients that can be addressed with the product is when I hear about the polyclonality. And let's just reflect on how exciting this is, that with the advanced melanoma and advanced cervical, in the former case, my understanding is that these patients have failed both PD-1 and PD-L1 therapies. Outside of these four walls, most physicians are extremely excited about what we might call classical immuno-oncology involving checkpoint inhibitors of that nature. But they themselves, they're not cheap, they're certainly not without side effects. Do you see any prospect of you marching up the standard of care to a point where you're competing with PD-1 and PD-L1 inhibitors, or even being used prior to their prescription?
Yeah, so right now, if you look at our repertoire, basically all of our metastatic melanoma patients have failed PD-1. And 40 to 65% of patients, metastatic melanoma patients that are put on anti-PD-1, whether it's Pembro or Nivo, essentially fail at some point, either right away or they respond and then they become refractory. So all of our patient cohort has failed PD-1, and we still see this good clinical efficacy with our TIL product. That being said, we're still investigating. It's difficult to get a population of metastatic melanoma patients that is naive to PD-1 at this point, but we do have a small cohort in a basket trial where we are looking at joining up with checkpoints, where we're looking at patients that are naive to checkpoints, giving them TIL and then following up with checkpoint therapy. So I think we can exist in the environment. I don't think that we need to compete, because we can exist post checkpoint inhibition failure, and we could potentially we can work with checkpoint inhibitors. And that's really something that we're looking at in the pipeline down the road, is the unmet medical need of those patients that have failed all treatments, and also thinking that we would like to be much earlier line and in combination with checkpoints.
Yep, that sounds like a lot of clinical trials.
It is, and so we have, of course, our LM144 trial, where we have just our metastatic melanoma patients that have failed PD-1. Actually, most of them have been on 3 to 6 prior therapies. And then we do have another part of our clinical trial, a basket trial, that we are looking at naive patients from cervical, head and neck, and melanoma that are naive to checkpoints that we're then treating with TIL and checkpoint.
So BLA 2020?
Yes.
2 BLAs 2020?
So definitely with lifileucel M145 for metastatic melanoma, we're projecting the submission of the BLA in Q3 and Q4 of this year. We've also had the end of phase 2 meeting with the FDA, and they were very favorable on the single arm registration trial, and with our pre-existing cervical cohort and just expanding that cohort, we can go ahead with BLA submission. So we're going to prepare for BLA submission this year, and hopefully by the beginning of 2021, we're hoping.
So shipping drug in 2021?
Well, the hope is that once we submit, depending upon the FDA, that we get approval, but we're hoping that both BLAs will be submitted within 2020 to 2021. Our manufacturing facility is scheduled to be
rolling
complete.
Submissions?
Pardon me?
Rolling submissions?
Yes, basically.
And how many patients are you going to be treating in 2021?
So we just finished treating our last patient on our cohort 4, which is our pivotal registration trial for metastatic melanoma. We started in March of 2019, and we treated 75 patients between March of 2019 and January 15th of 2020 to complete the registration cohort. We're going to continue on the cervical a cervical trial to try to bring that cohort up to 75. We currently have reported about 27 patients on that, but those treatments are ongoing right now. We also have the head and neck cohort that's ongoing, where we need to increase the patient number. But we also have we've submitted some further INDs, such as for the CLL, which is a heme malignancy with our PBL product. We also have some selection strategies, PD-1 selection, that we're hoping that we can initiate clinical trials in the very near future.
Yep. Well, so that's 2 BLAs, new manufacturing facility, in-licensing gene editing, and an IL-2 analog as well.
As well as selection.
What else are you going to do?
Whatever. The options are endless, right? I think that one of the things that when you see about Iovance, and you think of cellular immunotherapy, you spoke to it at the beginning, is that they're waiting for that bell to ring with cellular immunotherapy. We thought a couple years ago, and we've been sitting around at Iovance, doing our jobs, trying to get to the point where we can actually treat solid malignancies. And so what we know is that there's no cellular immunotherapy product for solid malignancies. And we believe that we will be the first. But lifileucel is only going to be one component of what Iovance can bring to solid tumors. That is the hope. We want to expand into multiple indications. We want to continue to perfect our current product that has clinical efficacy so that we can see greater durability, greater persistence, and we can treat more indications.
Good luck. You've got a lot of work to do.
I know.
We're all waiting for that bell to ring. Thank you very much.